Hello, brilliant young minds. Welcome to today's science adventure. I am your teacher and will guide you into the fascinating world of cells, the tiny building blocks that make up every living thing, including you. Today, we are going to shrink down to the microscopic level and discover what makes life alive. We will learn how scientists use microscopes to explore cells, what cells look like, and why they are so important. So get ready to zoom in and explore a world that is invisible to the naked eye, but it's full of wonder. Yes, today we will learn about discovering the cell. So think about what do you know, according to your prior knowledge, that who coined the term cell and how did he discover it? Think about it. Yes, he was Robert Hooke. Let's go back in the time to the year 1665. A scientist whose name was Robert Hooke was curious about the world around him. He took a thin slice of cork and looked at it under a microscope. What he saw amazed him. Tiny compartments that looked like small rooms. He called them cells because they reminded him of the rooms monks lived in at a monastery. What he actually saw were the cell walls of the dead plant cells. But this simple observation was the beginning of something huge, the study of cell, which is later on known as cell biology. It's amazing how one curious look through a microscope changed science forever. Cells are incredibly small. Some are just 0.001 millimeters long. That is smaller than a grain of sand, you can say. Even smaller than the grain of sand. Just imagine about the size of a grain of sand. You can't see cells with your naked eye. That's why scientists use microscopes to see them. Even the tiniest cells has important parts inside it like nucleus, cytoplasm, cell membrane, and so on. To study these cells, we need tools that can magnify them, make them look bigger, so we can see their structure and understand how all these structures are working over there. So, which tools we basically use, students? We use light microscopes, that is also included in your syllabus. What is basically a light microscope or how we can define it? A light microscope is a tool that helps us see very small things like cells. It uses light and lenses to magnify the image. How it uses light and lenses to magnify the image, we will discuss it later on in detail. When you place a thin specimen on the stage, there is a stage, and I will discuss the different parts of the microscope with you. So, the stage is the platform where we are going to put our specimen over there, the thing which we are going to observe and shine light through it. It can be reflected by means of the mirror, which is installed in some sort of light microscopes, as well as it can be shined directly through any light source which is already built in some light microscopes. So the microscope bends the light and makes the image bigger after reflecting. This lets us see details that we could never see with just our eyes, by using our naked eyes. Microscopes are the tools which are used to magnify the objects, to see them very clearly. They help us study everything, like from onion cells, cheek cells, to blood cells, to any tiny bacterial cell, even. Let's take a closer look at the parts of the light microscope. Each part has a special job. These are the different parts of a light microscope. You can see on your screens. I have just placed different names of these parts and as well as mentioned the diagram over there. So according to this diagram, the first one is eyepiece, then the next arrow is coarse focus, after that it is a fine focus, then objective lenses are there, rotating nose is there, after that there is a stage upon which you are going to put on the specimen, aperture is there, as well as mirror is installed in this diagram the given in your slides. Moving next, all these parts are now you can see on your screen in the labeled form. So I'm just going to explain each and every part with its function basically. What is the basic function of all these parts? So starting from your eyepiece, this is where you look through and it usually magnifies the image 10 times greater, 10 times more than its original size. The next part is objective lenses. These are found on the rotating nose and there are different magnifications are installed over there. They magnify the image even more, usually like 4 times, 10 times, 40 times, 80 times, 100 times. After that, you people can clearly have a look on the stage. This is a flat platform where we are just going to place our slide of the specimen which we need to look through. Then light source is over there. It shines light through the specimen so you can see it clearly. And there are two different knobs over there. One is focus knob and the coarse knob. So these knobs help us sharpen the image. There is a coarse knob for big adjustment and fine knob for tiny ones adjustments. Like by rotating the coarse knob, we are making the adjustment at a bigger scale. Is it clear? And the fine knob make it finer, make it clear, make the image clear to us. All these parts work together to help us see the tiny world of cells. So how to use a microscope in detail, step by step. You people can see on your screens now. Using a microscope is like learning to ride a bike, you can say. It takes practice. The more you practice, the more you have efficiency to deal with it. So first step. What is the first step? You just have to place the slide on the stage, then secure it with clips which are present. We are just these clips are there just to make them fix, stop it from any movement. Then we just have to turn on the light source if there is a light source in the light microscope installed over there, and if there is no light source then we just have to fix the mirror for reflection of light so the specimen is lit from below. So we just start with our lowest objective lens. Firstly, it is like 4 times. We just started from 4 times, and if the image is not clear then we use the next magnification, that is of the 10 times, and after that it will be of 40 times. But we use these magnifications one by one by rotating the objective nose over there. We can adjust the objective lens. So look through the eyepiece and slowly turn the coarse knob until the image appear to you. Because firstly when we are just going to look through the eyepiece, we are unable to see any kind of image over there. But after rotating the coarse focus, the knob, the bigger knob, which is known as coarse focus knob, so it will become clearer the image to you. After that, we use the fine focus knob to make the image more sharp and clear to you. And when the image is just going to be clear, and if we want to zoom in more, we just have to switch towards the higher objective lens. Like if firstly we were using 4 times objective lens, then we will just switch towards like 10 times objective lens. And if we want a more zoom in or the more detailed picture, then we will just shift towards the 40 times objective lens, which is placed on the rotating nose of the microscope. Is it clear? Okay, this is the sorry, it is your third step. Then fourth is we are just going to be okay, now just be focused on your slides now. Firstly, you're just going to take the first step that I have just explained to you, that you people will have to turn the microscope on, then move the mirror to reflect the light up towards the eyepiece and check that where the aperture is wide open, from which the aperture from where the light is just going to pass through from the source towards the specimen. And then you just have to turn the revolving nose piece so the lowest objective lens is pointing directly down. Is this thing clear? After doing this, we are just moving towards the next step, that place your specimen onto the stage and fix it with the help of clips. Look into the eyepiece and slowly rotate the coarse focus to bring your specimen into your focus. I have already explained all these steps in detail. After that, you just slowly rotate the fine focus until you obtain a clearer image of your specimen, and examine and explore what specimen holding still or moving in the slide slowly with your thumb. We can do it like we have two options. We can only move our coarse focus, or to get the specimen in our focus, we just have to move our slide, so it will be just directly in contact with those objective lens which we people are going to be used. What up next? Once you people have seen enough switch to the medium and then towards the higher objective lens, use the fine focus to bring the specimen into the focus as it is necessary. I have already explained all these steps in detail. Now, I just want to have a quick check. Are you able to answer these questions? What is the purpose of fine adjustment wheel on the microscope? Yes, try to find out the answer. What is the purpose of the fine adjustment wheel? Fine adjustment means fine focus. What is its main purpose? So what do you think? Yes, to magnify the specimen, to illuminate the specimen, to bring the specimen into sharp focus, or to change the lens. So which one is the correct answer for it? Think about it. Yes, exactly. The third option is the correct answer, to bring the specimen into sharp focus by means of fine adjustment wheel. Okay, so we are just moving towards another simple activity. You just have to trace out the path of light from it. So look at the diagram of the microscope shown here. By using your finger, or track the path of the rays of the light from the source to the eyes of the observer. In a line, you can also draw it over there. So think about that, how the line can be drawn over there. Light moves from which part? Like the light source or the mirror is over there, so the straight line from the mirror or from the source through aperture, it hits the specimen which is placed in the form of slide over there on the stage. After that, it moves towards your objective lens. Then through this knob, it moves towards your eyepiece, and as your eye is upon your eyepiece, because you are using this microscope, and it enters into the eye and makes the image on your retina. That is why you people are able to see that image. Have a look clearly in next how this light is just moving on. Light ray go through the aperture to the specimen, to the objective lens, then to the eyepiece and into the eye. But remember, light rays bend as they pass through the lenses, allowing the image to be magnified. This is not in the form of straight line like you people are able to see on the screen. It is just, I have just draw it in the straight line, but it doesn't mean it's always moving in the straight line, because due to the presence of lenses in it, it just have to bend to some sort. And the main idea of a light microscope is for the light rays to go through the specimen. So specimens must be thin enough for the light to pass through, must be thin enough. The specimen which we people are just taking to observe, it must be very thin so that light can easily pass through it. So the summary of all the lesson that we have discussed yesterday as well as today: what are cells and how does a microscope work? Yes. What are cells? A cell is the smallest unit of living organisms, and organism may be made up of one cell or many cells. Like organisms can be a unicellular or multicellular. And how does a microscope work? A microscope magnifies the structure of the specimen. Specimens must be brought into focus to be viewed, and it is done by moving the coarse focus as well as the fine focus. That's all we people have learned in these 2 days. All of you should be very well prepared, and your home task is you people just have to draw and label light microscope and then label it. And always and also you people will have to give one sentence about what each part of that microscope work, or you can say the function of each part of microscope. What you have to do, you people will have to draw the light microscope in your notebooks, and you will have to write the functions of each part of microscope. Okay, thank you so much. Stay blessed. Allah Hafiz.